I hope you are doing well. Before we start today's lecture, I want to say one small thing. I am not feeling well today and I have a fever, so if my voice sounds a little different, please excuse me. I did not want to delay this course because many of you are learning with me step by step. Thank you so much for your understanding and support. Now let's begin. Welcome to episode 9 of our NGS Bioinformatics Master's course. In our previous episode, we learned this basic concept of molecular biology, DNA, RNA, gene, chromosome, genome, and next generation sequencing. Now we are starting the practical part of this course. Today's lecture is very important because every NGS project starts with raw sequencing data. In this video, I will show you where to download real sequencing data, how to understand an NCBI SRA datasets, what a FASTQ file is, what quality score and thread quality score mean, and finally how to generate a FastQC report. So let's get started. First, open your web browser and search for NCBI. NCBI stands for National Center for Biotechnology Information. It is one of the most important websites for every bioinformatician and life science researchers. NCBI provides many biological databases including DNA sequence, RNA sequence, protein sequence, genome, research article, and public sequencing datasets. Today we will use the SRA database. SRA database. SRA stands for Sequence Read Archive. It stores raw sequencing data submitted by researchers from all over the world. In the search box, type the topic related to your research. For example, today I search for lung cancer for RNA-Seq data. You can search for any disease or organism such as breast cancer, lung cancer, diabetes, Alzheimer disease, microbiome, or any topic related to your research. Now you can see many datasets. These results show important information like the study title, sequencing platform, accession number, number of sequencing read, and file size. This information helps us choose the correct dataset for our analysis. Now let's open one dataset. This page contains detailed information about the sequencing experiment. At the top, we can see the study title, which tells us what the research is about. Here we can also see who submitted the data. Next, check the organism. This tells us which species was used in the experiment. Always verify the organism before downloading the data because different organisms require different reference genome during analysis. Next is the sequencing platform. Here we can see the instrument used for sequencing. Then we have the sequencing strategy. If it say RNA-seq, it means RNA was sequenced to study gene expression. You can also see whether the data is single-end or pair-end. If it is pair-end, we will obviously get into FASTQ files, read 1 and read 2. Finally, look at the run accession number. Obviously starting with SRR or ERR. This accession number is very important because we use it to download the sequencing data. Now we are on the European Nucleotide Archive, also called ENA. ENA contains the same sequencing datasets as SRE. Many researchers prefer ENA because it provides directly download link for FASTQ file, and instead of during command line tool, we can simply download the FASTQ files directly from this page. This is especially helpful for beginners who are just starting bioinformatics. Now let's open the FastQ file. This is the first file we receive after sequencing every NGS and also start with this file. Let's understand its a structure. Each sequencing read always contains four lines. The first line start with the at the rate symbol. This is called the read identifier. It gives a unique name to every sequence read. The second line contains the DNA sequence. You can see the letter TTGT. These represent the nucleotide sequence generated by the sequencing machine. The third line contains only a plus sign. It simply separates the DNA sequences from the quality information. The fourth line contains the quality score. The fourth And now, what is a quality score? A quality score tells us how confident the sequencing machine is that each nucleotide has been identified correctly. If the quality score is high, it means the machine is highly confident that the base is correct. If the quality score is low, there is a higher chance that the base may be incorrect. These quality scores are called Fred quality score. The Fred quality score is a standard way of measuring sequencing quality. A higher Fred quality score means a lower probability of sequencing error. For example, quality score 20 means about 99% accuracy or approximately one incorrect base in every 100 bases. If quality score 30 means about 99.9% accuracy or approximately one incorrect base in every 1000 bases. That is why sequencing data with quality score 30 our higher is generally considered to be high quality. Remember one simple rule. Higher Fred score equal better sequencing quality. And now before starting any analysis, we need to check the quality of our FastQ file. For this we can For this, we use a software called FastQC. FastQC, one of the most commonly used quality control tool in bioinformatics. It does not change the sequencing data. It simply checks the quality and generate a report. We are Here we can see the basic statistics. This section show information such as the file name, total number of sequence length, and GC content. On the left side, you can see different quality modules. Each modules check a different aspects of sequencing quality. For example, per per base sequence quality tell us whether the quality of bases is good across the entire read. Other modules check GC content, sequence length, distribution, adapter contamination, and overrepresented sequence. These reports help us decide whether the sequencing data is suitable for downstream analysis. Don't worry if these graphs look difficult today. In the next episode, we will understand each FASTQ graph one by one in a simple and practical way. So today's we are learned. Today's we learn what is NCBI, what is the SRA database, how to search and understand a sequencing data set, how to download raw sequencing data using ANA, what is a FASTQ file, why every sequencing read has four line, what our quality score, what is a flat quality score, and how to generate a FastQ report. These are the first practical step in every NGS bioinformatics project. In our next episode, we will explore the FastQC report, in details and learn how to interpret every graph like a bioinformatician. If you found this video helpful, please like the video, subscribe to SaraBioinformatics YouTube channel, and share this course with your friends and colleagues. Thank you so much for watching. And if you have any doubts, so please write in the comment box. See you soon in the next episode. Happy learning.